Hello friends, welcome to Biotech Guide. In this video, we'll break down the ICAR JRF 2020 Agriculture Microbiology section Core Crop Science paper with the simplest explanation. So, question by question, let's decode the previous year paper and boost your preparation in the smart way. Let's start. So part B, Core Crop Science 3. The number of questions are 50 and the mark is 200. Each contain 4 marks. So let's start first. The translation process occurs in association with which one of the following? So the answer is ribosome and RNA. As we know, the translation process where tRNA is bound with a subunit of a ribosome and the translation process occur. Then ER seers produced 21 monosomics in which one of the following wheat varieties? So the answer is China's spring. Okay. ER seers was a renowned geneticist who made significant contribution to wheat cytogenetics. Okay, so the answer is Chinese spring. Then name the small cDNA sequence that represent a unique segment of an active gene. SNP, snRNAs, ESTS or contigs. Okay, so SNP is, as we know, small nucleotide polymorphic DNA. So that is not right answer, is used for the only the mapping procedure or polymorphism. Then snRNA is also applicable in the RNA splicing procedure or mRNA interference. Then contigs are the small fragment of the DNA which is made by the two or many more kind of genes. And the third, ESTS is the right answer. EST's full form is expressed sequence tag. Expressed sequence tag is a short subsequence 200 to 800 base pair generated by sequencing cDNA. Okay. Then which one of the following species of Cajanus is used to introgress salt tolerance in the cultivated species of a Cajanus? So the answer is Cajanus scarabidus. Okay. Wild species of Cajanus used to increase the salt tolerance in a cultivated pigeon. Then under IPR, what protects the intellectual property created by inventors? So the answer is patent. Then the world's most inventory of the global conservation status of a biological species is being maintained by IUCN. Full form is International Union for Conservation of Nature. Here WWF is a worldwide fund for nature. It basically supports the cause of a world. Then MAB, Medicinal Plant Board on Mapping Initiatives, not related to global species. And IBWL, Integrated Protected Wildlife List, not actual related to this. Okay, so the answer is IUCN. Then met the following. So here PEDCON is related to this. GM is tunicata. Okay. So 1 is linked with a 3 here. Then popcorn is related to the everta, that means B1. Okay, so the answer is this. Then C is linked to the 2. GM is seritina, or flint corn is related to the GM is induta. These okay, so these are the classification are based on the genetic studies and the classification of a maize, races and a variant. Then another method the following. So the species or the number of a chromosome is given. So linium uctisium having a 15 number of a chromosome, so A4. Okay, this is the answer. Then B3. Okay, and the C1 18 and a D2. Okay, so this is the answer. Then another method the following. Crop and isolation distance for the certified seed production. So here hybrid rice having a 400 meter. Okay, A1. Then B Johnson grass 800 meter. Then sesame is a 100 meter and carrot 50. Okay, so this is the right answer. Then adventive embryony is example of vegetative propagation. Is a type of apomixis in which embryos arise directly from the somatic cells. Okay. Then loss in a vigor due to inbreeding is more visible in cross-pollinated species. Why? Because the inbreeding depression refers to a loss of a vigor, fertility or a yield due to repeated inbreeding. So the answer is a cross-pollinated species. Then if two individuals with a genotype AABB are crossed, what proportion of the offspring will be genotype AABB? Okay, so here they are asking for the recessive offspring small a small b. So here we calculate when we cross AABB cross AABB. So what we'll get? So when we cross A to A cross A, so possible offspring was what? AA, then capital A small a, another capital A small a, and at the end small a small a. So chance of getting recessive is 1/4. Similar, when we cross B into B, so we'll get capital B, then capital B small b, another capital B small b, and at the end small b. So chance of getting small b is what? 1/4. So when we multiply this 1/4 and into 1/4, we'll get 1/60. So the possible proportion of offspring genotype is what? 1/16. Then requirement for marker assisted backcross breeding are tightly linked marker with a target gene. Okay, so MABB, that means a marker assisted backcross breeding, is the transfer a specific target gene like a disease resistant or a drought resistance from a donor parent to elite variety. Okay, these are not relatable. Then chi-square test has the following application. So the answer is a test of segregation. Why? Because the check whether observed results match expected ratios such as the Mendelian inheritance parents, then monohybrid, dihybrid process, expected segregation ratio. These all are investigated by this method, test of segregation. Remaining are not relevant or defined use in the concept. Then requirement of AFLP marker are basically we required a restriction enzyme, adapters and a PCR. These three ingredients are required for the amplified fragment length polymorphism. Random primers are used in RAPD. Okay, and short sequence repeats are not related to the field. Then for breeding high quality chapati, wheat in India is preferred. So the Triticum asativum is a scientific name of a gehu. Then evidence for a DNA as a genetic material was obtained in which one of the following experiment? So the experiment is every every macroid, McCarthy, and another experiment is Hershey and Chase. Okay, so answer is A and E. Okay, Griffith controlled the transformation. Okay, so Griffith is not involved. These are not related, and Watson and Crick gives a double helix structure. Then a population consisted of a following genotypes in given numbers: 160 heterozygous, 20 homozygous recessive, 320 homozygous dominant. So what would be the frequency of a dominant allele? So here first we are doing what? We total the doing total. So we are having a 500 total genes. Now alleles are into 2, so 500 into 2, that means 1000 alleles. Now what they are asking? Dominant allele. So dominant allele always expressed via heterozygous and homozygous dominant. Okay, so homozygous dominants are 320 into 2, because in homozygous dominant AA both allele are there. Okay, so into 2 we calculate into 2, so 640 we get. Similar in heterozygotes, only one dominant allele is present, so 160 into 1, so we get 160. Okay, so total what will get? 800. Now we will calculate percentage. So 800 divided by 1000, so we will get 18%. 18% of the dominant allele. Similar we will calculate of a recessive allele. So recessive allele is expressed percentage. So 20% is a frequency of a recessive allele. Now the non-random association of a allele at a different loci in a given population is referred to as a linkage equilibrium. Is the non-random association of alleles at a different loci in a population. Then non-Mendelian inheritance include maternal, not a maternal but epigenetic and extranuclear inheritance only. Okay, third option. Why? So maternal effect is the phenotype of a offspring is determined not by its own genotype but the mother genotype, and extranuclear inheritance supposed through a organelle like a mitochondria and a chloroplast which have their own DNA. Okay, so answer is 3. Then Morgan discovered a linkage when studying two genes in which one of the following Drosophila. So the answer is X chromosome. X chromosome of the fruit fly Drosophila melanogaster, that certain traits were inherited together more often than expected by independent assortment. Then Carey Mullis invented PCR and received a Nobel Prize in chemistry. Then the antibiotic resistant genes are mostly used as what kind of marker? So the answer is a selectable marker. They allow researchers to select transformed cells from a non-transformable ones. Then the molecular is used for the mapping. Scorable and countable are not relatable. Then expanded RAPD, random epilymphoid polymorphic DNA 4. Then hybrid between plasmid and a phase lambda chromosome are known as a phosmid vector. Here plasmid only derived from the plasmid. Then binary agrobacterium derived vector is defined as a binary vector, and cosmid define a different hybrid plasmid plus a cos sites of a lambda phase. Then DUS testing is necessary for what? DUS stand as a distinctness, uniformity and stability is essential for the certification of a variety. Certification of a variety is needed. C only. Then CSTL is located at Varanasi. CSTL is a central seed testing laboratory. Then two-line system of hybrid seed production is possible in TGMS. TGMS, that means a thermosensitive genetic more sterility, is a process, a two-line hybrid seed production system where a male sterile line whose sterility is controlled by the environmental factors and a fertile pollinator line. Then a seed multiplication ratio of a paddy is what? 1:80. That means a number of seed produced from the single seed. Like for paddy rice, typically seed multiplication ratio is 1:80, means 1 kg of a seed can produce 80 kg of a seed under the optimal condition. Now, which of the following are certified classes of a seeder under the Seeder Act? So, the foundation seed and the certified seed, those two are the mandatory. As per the Seed Act 1966, India certified classes of seeds are those that are officially recognized and go through the certification process to ensure quality and the genetic purities. So, formulation seed produced from the breeder seed and a certified seed from the foundation seed. So, here we completed.